Hello. Next we are going to discuss about a topic that is epistasis. What is epistasis? Okay. So let's discuss first what is epistasis. As you all know, epistasis is a non-allelic gene suppression. What is non-allelic gene suppression? I have already mentioned you. That is, if two alleles are present, then say for example, one allele is capital T small t, another allele is capital P small p. These two are known as non-allelic suppression. Here in case of epistasis, two pair of alleles are involved. And one of the feature of epistasis is that it will suppress both dominant as well as recessive non-allelic, which we have discussed in the previous class. Now we are going to discuss about epistasis in detail. That is, what are types of epistasis. First is the dominant epistasis, second is recessive epistasis, and third is duplicate, fourth is duplicate dominant, and fifth is duplicate dominant recessive interaction. What are the type, what are this, and what are the phenotypic ratio? We are going to discuss one by one. As you all know that the ratio of the hybrid cross is 9 is to 3 is to 3 is to 1. But we have seen that how due to gene interaction this ratio gets diverted and changed into another ratio. What are this ratio? We will be discussing, and what are the tick to remember it? We are going to discuss also that. So let's begin. First we are going to discuss about dominant epistasis. So as you have seen, the word dominant stands for the expressed this gene or the allele which expresses itself is known as dominant. So dominant epistasis means what? The dominant allele of one gene will suppress both the allele with of the dominant as well as the recessive one. Here for example, if we see here an example that is of a, let's say here the A, capital A is dominant over small a, as we know already. So these capital A is dominant allele of one gene, and these capital B and small b are another allele. So these capital A will suppress both capital B as well as capital small b of the another allele. This is known as the dominant epistasis. That is, the capital A will suppress both capital B as well as small b of another allele. Okay, this is known as dominant epistasis. Due to this, the phenotypic ratio will be 12 is to 3 is to 4. Okay, how is 12 is to 3 is to 4? Let's see. So here the parent one, here the purple color will be formed because both A is present. So remember, wherever A will be present, purple color will be formed. Okay, and here white color will be formed because there is no A pigment as well as B pigment. Okay, when we will cross both of it in the F1 progeny, we will get purple color. Why? Because capital A is present. Okay, when we self cross them, we get the F2 generation, that is 12 is to 3 is to 1. Why we get this ratio? Let's understand it in here. Suppose this is a white substance, and here the enzyme A is responsible for the purple product, and enzyme B, that is the B allele, is responsible for the red product. Here A allele is responsible for purple product, and here B allele is responsible for red product. But what happens due to epistasis, due to dominant epistasis, these A allele will suppress this B allele from getting expressed, as a result of which wherever the capital A will be found, there only purple color will be produced and the red product cannot be found. And wherever the capital A will be formed, purple color will be seen. So you can see here in the F2 progeny, we will get this type of progeny. That is, if you see here, we will get capital A, here capital A is present, so 1, here 1 capital A capital B, here capital B will be present. You can see here capital B is present, but due to capital A is suppressing this capital B, that's why it can't express itself. Okay, that's why here also purple color will be found. Another example here, small b is present, but capital A is suppressing the small b, that's why the purple color will be formed. So wherever you will see capital A, there purple color will be formed, because here the capital A is dominant over small b, capital B and small b. And here red color, 3 red color will be formed, because here there is no functional capital A. Okay, there is no capital A. Because of the absence of capital A, purple color can't be formed. That's where red color will be formed, that is 3. And 1 white color, that is small a small b. So due to this we get the ratio, that is 12:3:1. Okay. So by this we cleared that the dominant allele of one gene will suppress both the dominant as well as the recessive allele of non-allelic gene. So this is one type of dominant epistasis, that is dominant epistasis. Next, moving to the recessive epistasis. Here what happens? Here no one will suppress anyone. Here what happens? Here the individually capital A, that is the dominant allele of one gene, can individually express itself, but the dominant allele of another gene can't express itself individually. It requires the help of another allele. Let's see here. So here for example, capital A, that is dominant allele of one gene, it can express itself and can produce color if it is present alone. But capital B, okay, capital B, that is the dominant allele of another gene, can't express itself unless and until it is present with capital A. So these capital B can't express itself individually. This is known as recessive epistasis, due to which we get the ratio 9:3:4. How it is formed? Let's see. First we will cross the red color. Here first we will see how the red color is formed, because the white substance, when the enzyme A is given, here the enzyme A is responsible for the red product. Okay. So you can say that when A will be present individually, it can produce the red color. But if enzyme B is present individually, it will produce no color. Okay. When both enzyme A and both enzyme B will be present together, then purple color will be formed. So you can say that the B enzyme will express itself only in the presence of enzyme A, otherwise it can't express itself. So here you can see that here capital A, that is red color, can express itself. Okay, capital A will express itself, and here it as no dominant genes are present, so white color flower. Next, when we will that is when we will F1 generation, when we will cross them, then we will get the progeny, that is purple color, because of the presence of A and B. So both A and B will produce a purple color. Here you might be wondering why red color is produced, because here this capital A is dominant over this B. Okay, this both the capitals are A is present, that's why red color will be produced. But here only single capital A and small a is present, that's why it will here both the capital A and small capital B will produce the purple color. When we will self cross them, then we will get the ratio 9:9 purple, 3 red and 4 white. So wherever both capital A, capital B, capital A, capital B, capital A, capital B will be present, purple color flower will be formed. Okay, so we will get 9 purple. And wherever only capital A is present, you can see here only capital A is present, okay, or small a is also present, then red color will be formed. Okay, if capital A also present or small a will be present, then also red color will be formed. But white color will be formed in the absence of both capital A and small b. So 4 white color will be formed. That's why we will get the ratio, that is 9:3:4 ratio. This is known as recessive epistasis. Next, moving to the duplicate recessive. Okay, here what happens, both the recessive allele will play the role, that is recessive allele of gene 1 as well as recessive allele of gene 2 when present together will express certain color. If it is not present together, they can't express its color. What say? Let's see. Suppose a gene pair of purple dominant over white, we know that, and in pair gene B color dominant over white. So what happened? We get the ratio that is 9:7. So wherever you will see the ratio, that is wherever you will see small b, here also, wait a minute, okay, sir, here small b, small b, small b, small b, small a, small a, small a, small a, and small a, small a, small a, small a, small b, small b. These type will produce one type of color, okay, because here both small a, small b, small a is present, okay, that's why they will produce the white color. And here you can see small b is present, but it is present in single condition. Here also small a is present, but it is present singly. That's why they can't express themselves. So wherever they both the alleles are will be present, either of the allele, that is small a or small b, that is known as duplicate. That is both the allele will play the role, both the recessive allele will play its role. Then only the white color will be formed. Here the small a will be epistatic to capital B, and small b will be epistatic to capital A. Okay, as the recessive allele is being masking the effect of dominant allele, that's why it is known as duplicate recessive. And here we get the ratio 9:7. Next, moving to the duplicate dominant. Here what happens? The allele, that is dominant allele, when present individually as well as when present together, expresses themselves. Let's say, for example, if enzyme A or product gene A, that is dominant allele, is present individually, then also it will produce the color pigment. If B gene is also present individually, then also color pigment will be formed. If present both together, that is A and B, if present together also, then also the color will be formed. But if both the dominant gene or both the dominant allele will be absent, then white color will be formed. That's why we get the ratio here, that is 15:1. You can see here, that is, if we take an example of triangular shape dominant over ovoid and triangular dominant ovoid, you can see we get the ratio wherever capital A, capital B, and if individually if also capital A and capital B will be present, we get the triangular color shape. And wherever no absence of capital and capital B dominant gene, then small a, small b will be present, white color, that is one ovoid will be formed. So here the epistasis seen, that A is epistatic to B and small b, and B will be epistatic to capital A and small. So both the allele will be expressing themselves, that is both the dominant allele. That's why it is known as duplicate dominant. Next, moving to the last part of the epistasis, that is dominant recessive interaction. Here what happens? Here the product gene A is responsible for certain color pigment, that is Moldavian color of the wheat color, that is kernel color. But what happens? This product gene B will suppress the product A, as a result of which the color can't be formed. So wherever the B pigment B gene will be present, there the color can't be formed. And wherever the capital A is present individually in the absence of capital B, there the color will be formed. So you can see here, that is we get the ratio 13:3. Okay, 3 for here 3 represents the where A is present individually and 13 represent the colorless. Okay, 3 represent here the colored form and 13 represents the colorless form because of the presence of gene B, because the gene B masks the effect of gene A, which is responsible for color production. So this is known as dominant recessive interaction, because the B is dominant, and this is also known as dominant suppression also, because here the dominant gene B is suppressing the effect of gene A. So these are the types of epistasis, that is, first is known as dominant recessive interaction, then is the duplicate dominant, duplicate recessive, recessive epistasis, and dominant epistasis. Now we will remember the trick, how to remember the trick of this ratio. Okay, wait a minute. Okay, here I am writing, you know that the ratio of 9:3:3:1 1. Okay, here this is the ratio of the dihybrid cross of the Mendelian cross. So remember that whenever you are writing about a dominant epistasis, then you need to add from here, that is 9 plus 3 is 12. Okay, so the ratio becomes 12 is to 3 is to 1. Okay, you can check here. Okay, 9 is to 3 is to 1. Okay, here sorry I have mistaken, I have written 4. 9 is to 3 is to 1. Okay, sorry, 12 is to 3 is to 1. Okay, 12 is to 3 is to 1 is the ratio. Okay, how it is found? Because of the, here you can add it up. Okay, by adding up this side. So 9 is to 3, 9 plus 3 is 12, and here as it is 3 and 1 will be written. So this is 1 ratio. Then while adding for the recessive one, you need to add from this side. So this side is for dominant, right hand side is for dominant, and left hand side is for recessive. So for recessive you need to add from this side. So the ratio for the recessive becomes 9 is to 3 is to 4. 3 plus 1 is 4. Okay. Now next, this is the dominant recessive. Now you need to add 2 more times when you add for the duplicate dominant as well as duplicate recessive. So let's see, first we will add for the duplicate dominant. So 9 plus 3 12, then 12 plus 3 is 15. So we get the duplicate ratio for the dominant is 12 is to 1. And for recessive you need to add from this side. So 3 plus 1 4, 4 plus 3 7. So we get the ratio 9 is to 7 for duplicate recessive. And for dominant recessive interaction we get the ratio, that is 13 is to 3. Why we get the 13 is to 3? 9 plus 3 12, 12 plus 1 that is 13 and is to 3. Here it is some different from that of the usual mechanism which we are following. But for dominant and recessive, this trick you can apply. So remember this 9 is to 3 is to 3 is to 1 ratio in the dihybrid cross. Whenever you are adding from the dominant side, add from this side, and if duplicate, then you need to add 2 times. Okay, same in case of recessive. When you are adding up for the normal recessive, 1 time, for the duplicate recessive you need to add 2 times. So remember this ratio. These ratios are very important because it is asked in the exam, as well as for the crossing over also. Whenever you are writing in the exam also, this will be very helpful. So this is all about the epistasis concept, and in the first part we have discussed about the gene interaction. So this is all for today. Thank you and have a nice day.